Hello, and welcome back. We're going to continue talking about DNA replication, all right? With the process of DNA replication, there are going to be a number of steps that need to happen in the process. We are going to need to separate the two strands of our double helix. Okay. We are going to have enzymes that are going to read our template strand. Okay. Now, the template strand is going to equal our parental strand. Okay. So it's going to be the strand of DNA that is conserved between copying, okay? So we've got enzymes, let me make that plural because we've got a lot of enzymes that are involved that are going to read the template, going to read the parental strand. And in the end, we are going to generate two identical copies. Of DNA. Okay. Now, one of the things we talked about last time, right? We talked about DNA polymerase. All right. That DNA polymerase is our photocopying enzyme. And for the most part, DNA polymerase is really, really accurate, but it's not perfect. It's not 100%. Okay, so that DNA polymerase has a proofreading function to it, okay? Because we don't want to make mistakes when we're making copies of the DNA, because when we make mistakes copying the DNA, that's how mutations can be introduced into DNA, okay? So what I have over here, I've started with our DNA replication fork, okay? And it's called a fork because you've got two sides to it, you know, just like you kind of come to a fork in the road. This is a fork in the DNA, all right? And what you're going to have happen with DNA replication, okay? On one strand, okay, on the strand that runs 5' to 3', you're going to have continuous synthesis, meaning the DNA polymerase is going to be able to read that strand. It's going to make the copy in one long, continuous process. Our other strand, our 3' to 5' strand, is going to be a little bit different. It's going to be discontinuous because DNA polymerase only goes in a 5' to 3' direction. So we're going to have to synthesize the other strand in pieces, all right? So that 3' to 5' strand and the discontinuous synthesis on that gives rise to Okazaki fragments. And I really should capitalize that O, because Okazaki fragments were named after the scientist who characterized them, okay? And this happened to be a Japanese scientist named Okazaki, all right? And I actually like saying Okazaki. It's one of my favorite science words, okay? So I'll talk a little bit about the Okazaki fragments. I'm going to start out talking about the continuous synthesis first. just because that's going to be easier, okay? So what we're going to have, we've got our DNA replication fork. We've got to have at the head of that fork our helicase, okay? You can kind of think of the helicase there working to unzip the DNA double helix, okay? So our DNA helicase is going to move in this direction, okay? It's going to move up our DNA strand. It's going to melt those hydrogen bonds between the base pairs to create an opening, okay? And in order to keep that replication fork open, we've got some specialized proteins that come in and attach to the sugar phosphate backbone of the DNA molecule. And those are single-stranded binding proteins. And your book will abbreviate that SSBP, and that's what that stands for, single-stranded binding proteins. So it just attaches to the DNA double helix, and having that protein there prevents our DNA helix from coming back together after helicase opens it up, okay? So we've got our helicase that's going to open things up. We've got our single-stranded binding proteins that are going to hold our replication fork open. And then we're going to have another enzyme, and I'll put it up here because I'm kind of running out of space. We've got topoisomerase, okay? That topoisomerase is going to prevent the twisting that happens, okay? Like if you try and pull a rope apart, it kind of bunches up further up on the rope. The topoisomerase, essentially what it does is it kind of nicks the sugar phosphate backbone so it can, that strand of DNA can spin, and it relieves the torsion or the stress that's on the DNA double helix from opening it up, okay? So that's our topoisomerase. It's going to go ahead of our helicase. We've got our single-stranded binding proteins, okay? The other thing that I have on here, and I've got this in blue just to distinguish, all right? The blue is going to represent our RNA primer, okay? And the RNA primer is put down or added to the replication fork via the RNA primase. Okay, so that RNA primase is just going to be a protein. Well, let me put it down here to put it on this side. Okay, be a protein that's going to put down a short strand of RNA in order for the DNA polymerase to come in, grab onto that, and start adding nucleotides, okay? Now in this diagram, I've already kind of filled in this first strand, all right? The green here, this is our new strand of DNA, all right? And what we'd have, what can I do? Let's make it a square up here. This is going to be our DNA polymerase, okay? So we've got our DNA polymerase that's going to continue reading our template strand, our parental strand. It's going to add in the new nucleotides. In this case, the next nucleotide that DNA polymerase is going to add would be a cytosine because we've got a guanine here. As helicase melts that double helix, it will continue to read up that strand of DNA and add in the correct base pair, okay? So that's why I kind of filled in the first one here. This is our continuous strand over here, all right? So this side over here is going to be continuous. This one's the easier side of things, okay? Because we lay down our first primer, DNA polymerase latches on, starts chugging away, filling in nucleotides, and it just can plug along until it reaches the end of the DNA molecule, okay? On our other strand, that's why I have this one still open. I'm going to kind of fill this in so you can see what happens, okay? We've got our RNA primer that's set down here. We would need to have another RNA primer down here to start laying down another bit of primer in order for that RNA polymerase to come in, all right? Or sorry, RNA polymerase, DNA polymerase to come in, okay? Our DNA polymerase is going to latch on to that primer, and it's going to start filling in. So it's going to start moving down our strand of DNA, and it's going to insert those new nucleotides. And that would be C. Okay? So it'll sit that in, and we're going to have, just so I can show this here, we've got our primer strand. There's going to be a kink in this one just because of how I'm drawing it. There wouldn't normally be a kink, okay? So we've got that, and then we've got our next strand of RNA primer. Oops. Okay. So what we have here, all right, we've got a continuous piece here, but because that DNA polymerase bumps up against the next RNA primer, that leaves a gap in our sugar phosphate backbone. That's where our next enzyme is going to come in, okay? Because we're going to have to remove this bit of RNA primer, okay? So I've been talking about DNA polymerase like there's only one DNA polymerase. And that's not entirely true. There are multiple DNA polymerases. In fact, there are 3, all right? They all have slightly different functions, okay? DNA polymerase 3 is going to be the one that's functioning here. DNA polymerase 1 is actually going to come in, is going to remove our bit of primer. It'll add in DNA, oops, DNA, okay? But what's going to happen there then is you've got a gap between the 2 strands of DNA, all right? To fill in that gap, this is going to be where DNA ligase comes in, okay? So in terms of DNA replication, there are multiple polymerases. At this level of biology, I'm not going to ask you to distinguish between DNA polymerase 3, DNA polymerase 1, although I think your book does describe them. What I want you to know is there's one DNA polymerase that's going to be responsible for primarily synthesizing the new strand of DNA. There's a different DNA polymerase that's going to come in and remove our primers, the RNA primers, okay? When we remove those RNA primers now, we've got a gap in that sugar phosphate backbone that we've got to seal up because we don't want gaps in our sugar phosphate backbone because that would make the DNA molecule weaker. So we've got DNA ligase that's going to come in. That DNA ligase is going to connect those 2 pieces of DNA, okay? Hopefully that makes sense, okay? The other thing I brought up, I talked about Okazaki fragments, and that Okazaki being one of my favorite words, okay? That short strand of DNA that's synthesized is going to be our Okazaki fragment, okay? The short bits of DNA that are on the 3' to 5' end, okay, that are synthesized discontinuously, those are going to be our Okazaki fragments. And what I should have filled in here, okay, so you can see how this works. We've got a 3' end here, so this has to be a 5' end. Remember, the DNA polymerase only works 5' to 3'. That's why this strand gets synthesized continuously. Here, we've got our 5' down here, so our 5' end has to be up here, okay? So this strand is synthesized discontinuously. That's what leads to the Okazaki fragments, all right? And the Okazaki fragments then are connected together by the DNA ligase, all right? So that's kind of a quick overview on DNA replication. That kind of shows you where the enzymes are that I want you to know. I want you to know that where they're located on the DNA replication fork, and I want you to know the function of each of the enzymes I talked about, as well as the protein, like the single-stranded binding protein, okay? Please be aware there are more proteins involved than what I've discussed here, but for this course, these are the ones I want you to know, all right? If you have any questions, please let me know.